reach to the third and the final part of the replication, or the third requirement of the DNA replication, and that is the extension of the primer. So this extension of the primer will be done by special enzymes that are called as the DNA polymerase. So if we revise what we learned until now, the first thing is the DNA must be denatured. That is done by providing high temperature. Then the gene which should be replicated must be tagged with the primer. So primer is attached. So we have learned that primer, one job of the primer is that it will be specifying that which gene should be replicated. So primer is attached. So now you can imagine the two strands of the DNA are separated. So there will be two primers that are attached, one on one strand and the other will be attaching to the other strand. Both the primers will have different number and sequence of the nucleotide, but what will be common, both these primers will be RNA primers. So they will be attaching at specific site and they will be giving a signal that okay, this is the gene that should be replicated. And what is this signal for? This signal is for the DNA polymerases, and DNA polymerases are those enzymes that are required for the process of replication. Remember, we discussed the nomenclature of the enzyme. So if you focus on the name, the polymerases shows they are family of enzymes. The second word is polymer. So what is polymer? Generally, whenever something has a basic unit, we consider it as monomer. Like protein has a basic unit, which is amino acid. So we say one amino acid is one monomer. Mono means one, mer stands for body unit. Monomer means one unit. So if you attach many amino acids together, this will become a protein, and protein is also considered as polymer amino acids. Poly means many, mer means units. So it means that this enzyme, function of this enzyme is to make polymer, to make units combined together. And what are we talking about? We are talking about replication. Replication also has a DNA, also has a basic unit, which is nucleotide. So it means that these are the enzymes that combine the nucleotides, and the third word shows the substrate of whom nucleotides, DNA nucleotides. So now we come to know that DNA polymerases are the enzyme that can join the DNA nucleotides together. And we know this joining of the DNA nucleotides together is the process of replication. But there are certain things that we must know. A DNA polymerase has certain requirements. First requirement of the DNA polymerase is it needs activation. Remember, it's an enzyme. Enzymes are not active all the time. So at the time of replication, you have to activate it. Now, how to activate DNA polymerases? They need specific pH, they need specific temperature, they need specific chemicals for their activation. This you will learn in the cell and molecular biology. The first requirement of polymerase is activation. The second requirement is DNA polymerase need a template. You show them a DNA strand and they will make the missing part. Remember, DNA replication is semi-conservative. You need one strand and you can make the missing strand. So this is the requirement of DNA polymerase, that you have to show them one strand and they will make the other strand. So first requirement is activation, second requirement is template. The third requirement is that DNA polymerase cannot start the process of replication. Right? DNA polymerase cannot start the process of replication. They can continue the process of replication. So now we can understand why the primer is attached, because primer has not only tagged that this protein need the process of replication, but also they have provided a start. That is why the word primer means starter. So, and another important thing is that DNA polymerase, it always needs a 3' to continue the process of replication. So it is very interesting. It means the process of replication is not random. The process of replication is directional. The DNA polymerases are coming to some specific direction and then they will be continuing. This I will explain in a bit of more detail in the next slide, where we have shown certain image to explain the process of replication. But at this slide, always remember that the process of replication is directional and the process of replication will be in the direction of 5' end to 3' end. What does it mean? I will be explaining on the next slide. But three conditions of the DNA polymerases I have mentioned. First, DNA polymerases need activation. The DNA polymerases, they need a template to whom they will make a copy. And then DNA polymerase always need a primer, because the primer will initiate the process of replication. They will tag a gene that this should be replicated, and then they will provide a free 3' end from where the DNA polymerase will continue the process of replication. How? We will be discussing on the next slide. So this is a simple example to explain the process of replication, and more specifically to explain the function of the DNA polymerase. First, understand the color codes, then it will be easy for you to understand. On the left side, we have just shown the colors. First part is showing a double-stranded DNA which is stable, and the next part is unstable. This gives you the concept that many genes are present on one chromosome, but they all do not unstable at the same time. Those genes having different composition, they will behave differently. So you can see one part of the DNA is stable, cannot be used. The next part is unstable, so a zipper-like structure is present which is showing that this DNA has been denatured. Then to this structure, imagine, you can see there are blue color primers they have shown. So two strands are separated and there are blue color primers that are attached with one another, and then each primer is extended by the DNA polymerase. So you can see one primer, one side is making its missing part, right? So now come to the figure that is present on the right side. It is the same figure, but it is showing the DNA nucleotides, because actually DNA is made up of nucleotides. So here the nucleotides are shown. Important thing is, on this figure I have shown the 3' end and 5' ends, right? So just focus on now the right hand side of this figure. You have one strand of the DNA which has starting from 3' end ending on the 5' end. Similarly, you have the other side starting from the 5' end ending on the 3' end. The two strands are anti-parallel. Now nature has picked this gene for replication. So on the right side, on the 3' end of the DNA, a primer is attached. On the second strand of the DNA, a second primer is attached. See carefully, both the primers are attaching toward 3 to 5 side of the DNA, right? So if the primer is attaching to the DNA, the primers are also anti-parallel. Just like two strands of the DNA are anti-parallel, remember the base pairing thing, I had shown my hands to attach to thumbs. If the two strands are anti-parallel, obviously the primer should be also anti-parallel. It means the first nucleotide of the DNA, if it has a 3' end free, the primer when it will be attaching, its 5' end will be free, right? So similarly, all the nucleotides of the primer, they are attached with one another. Imagine in your mind the attachment of the nucleotide I had shown you on the board. I had drawn two nucleotides, sequence of the two DNA strands I had shown you on the board and had connected them in an anti-parallel manner. Similarly, the primer will be attached in an anti-parallel manner. Now the primer can be 15, 16, 17, 18, up to 30 nucleotides. So all these primer nucleotides are also attached from third position and fifth position with one another. So you can imagine that the first primer nucleotide has a free 5' end. So a time will come, a point will come where primer will finish. So the side of the primer, which is the last nucleotide, will have a free 3' end. Remember the last slide, the requirement of the DNA polymerase is free 3' end. If you give them a 3' end, they can make a bond with the next nucleotide on the 5' end side, right? So in this way, nature has put a blue color primer, and this blue color primer has a 5' end and 3' end. So the last nucleotide of the primer has a vacant 3 position, and when the DNA polymerase will come, it will see, okay, this is a single-stranded DNA, okay, there is a primer attached, so the process is initiated, and it will see that a 3 nucleotide is available. For example, in this example, you can see that the last nucleotide of the primer is A, and it is joined with the T of this example, and the next nucleotide. So what DNA polymerase will do, it will look for a C nucleotide, because guanosine bind with the cytosine nucleotide. So it will look for the cytosine nucleotide and attach like a usual 3 to 5 attachment of the nucleotide. So when the first DNA nucleotide is attached with this structure, then what will happen? This DNA nucleotide again has a 3' end, which is free, because the 5 position of DNA nucleotide is attached with the 3 position of the primer, so its 5th position is vacant. So now the DNA polymerase will again look for the next nucleotide, because it needs a template. It will see, okay, next is the C nucleotide on the DNA. It will look for a G nucleotide and it will bind it. So every time the primer will be providing, the DNA nucleotide will be providing a 3' end, and the DNA polymerase will be binding a nucleotide toward the 5 sides. So 3 and 5 side will be bonded with one another. The 3 side will be vacant again, and the process will go on again and again. You may have studied in previous classes that the DNA polymerases, some of them can only undergo replication, some of them can undergo the process of proofreading. So they will come back to see if all the connections are made properly. And when we had discussed about mutations, we had discussed that the DNA polymerases, after extending the template, after completing the process of replication, they go back and they check if there is some error, they remove it. That is why the DNA process, application process, is very much conservative. So in this case, what happens that when the DNA polymerase continue the process of replication beyond the primers, in the meantime, the enzymes, they remove the primer as well. Remember on the last slide I mentioned the DNA and RNA interactions, they are weak. So they are just temporarily attached so that DNA polymerase continue them the process of replication, and in the meantime the primers are removed. So when the DNA polymerase will complete the process of replication, and obviously it will keep on adding nucleotides until the stable DNA part reaches, because the genes after this region are again stable. So once the DNA replication is complete, the polymerase comes back and it sees, oh, the primer is gone, there is some gap available over here. So the DNA nucleotides will be filled in this gap so that the whole place becomes one unit again. So in this way, on one strand of the DNA, it is on the 3 to 5 side that a primer will be attached, and DNA polymerase will be start running from 5' direction to the 3' direction to make a copy. same thing will be occurring on the other strand of the DNA. So in this way, when this polymerase will finish their jobs, there will be two copies of the DNA that are exactly the same. So this was what we have learned today about DNA replication and the process of regulation at the DNA level. We have learned that all the genes present on one decondensed region of the DNA will not be entering into replication at the same time. We also learned that there are many different DNA polymerases that are available, and why there are different polymerases. All are doing replication because we have different genes. The composition is different, so for different gene composition we have different polymerases. Some DNA polymerases can only do replication, some can do replication as well as proofreading, while some can only undergo exonuclease activity or the cutting of the nucleotide activities as well. So the basic concept that I want to give you is the process of replication is diverse. All the genes do not undergo replication at the same time, and that is the basic of temporal gene expression, because all genes are not working at the same time. They will not give rise to protein at the same time, and that will give us diversity, the different phases of the cell cycle. So I hope you found this discussion interesting one. Maybe you found something new, some new points in this discussion. So keep discussing with one another, and if you have some questions, come up to me. We can discuss further in the group so that we can make the things clear. So inshallah we will be moving to the next level inshallah soon. We will be talking about regulation at the level of RNA. Until that time, stay at home, stay safe, and make your time constructive by discussing these different lectures and presentations with one another, and come up with questions. Until next discussion, I wish you the very best of luck.